Hello and welcome to the Amplifying Scientific Innovation video podcast. I'm your host, Dr. Sophia Ononye, founder and CEO of the Sophia Consulting Firm, a life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast is to showcase scientific innovation stemming from global life science companies and the life science industry at large through conversations with senior leaders who share their unique leadership journey, corporate vision, and industry outlook. My guest today is Sam Michael, the Chief Information Officer and Director, Automation and Compound Management at the National Center for Advancing Translational Sciences, NCATS, and Dr. Mark Ferrer, Director of NCATS Intramural 3D Tissue Bioprinting Laboratory, a multidisciplinary group that produces, validates, and uses 3D biofabricated tissues for disease modeling and drug discovery. My interest in 3D bioprinting and the field of regenerative medicine led me to reach out to Mark and Sam. I am truly excited and honored today to have my first set of interviews with the National Institutes of Health and to advance public awareness of the mission and objectives of the National Center for Advancing Translational Sciences. Welcome to the show, Mark and Sam.
Thank you, Sophia. Thank you for having us.
It's my pleasure. So I always start this podcast the same way with what I believe is one of the most important questions I'll ever ask anyone, which is, what is your definition of scientific innovation? We'll start with you first, Sam.
No, it's a great question. And so I think scientific innovation it's important to note you don't always want innovation for innovation's sake. And I think something that's really important is a key aspect of something that both myself and Mark have worked on for years is we have many things which are operational. And so sometimes you want operational excellence because things don't necessarily want to innovate a process. They want a process just to run smoothly. And I also think that that's a direct tie-in sometimes to innovation because you have to understand what is operational and know it and master that, but then that's where you face the limitations. And so when it comes to 3D bioprinting, and part of our history of this at NCATS is we had operationalized 2D cell structures and 2D screening for many, many years. And just for perspective at NCATS for high throughput screening, since 2005 to today, we've run over 1,000,01536 well plates. Wow. So a huge number. And of those numbers is what we saw essentially were that about half of those were cell-based assays and traditionally 2D cell-based assays. And so this is where innovation to me is taking a process and then understanding the limitations. And what we saw despite all of this screening is we weren't necessarily getting better results. And so the question wasn't that could we operationalize this? We could, is rather was maybe there were a problem with the model. And then this is where we can hand this over to Mark because we saw there was a need for innovation because the models we were using just didn't have the accuracy that we were hoping for. And that really spawned. And so back to answer your question, it's sort of, I always view it from where do we face our operational limits? And then once we encounter those limits, what are we going to do and what are we going to invest in to see if we can actually innovate to overcome those barriers?
Wow, extremely well said, understanding and extending operational limits. And that comes with scientific discovery itself and the process of, staying committed to doing something and to elevating the state of the science. So thank you for sharing that. And Mark, I'm curious to know also, what is your definition of scientific innovation?
No, in this sense, I'm very much like with Sam. I think I view scientific innovation not as almost a process, where you have to identify and recognize a problem, and then you use new ideas and new concepts, new scientific ideas and concepts, and you apply them to solve this problem. But I think what's important and what Sam is saying is it's not a linear process, right? It's almost like an iterative, instead of back and forth process. So you might have a problem that you want to solve, and then it's like, what idea am I going to use? What new technology am I going to use to solve that problem, right? Sometimes you have ideas and you have technologies and you don't know what to use them for, right? And you might have to think about what can I use this? What process I can help with this new idea? And one of the things that Sam mentioned, the whole concept of operation, right? And making it happen. Sometimes you might have ideas and you have a problem, but to go from one to the other, you really have to implement and the aspect of operations and implement ideas towards solving a problem, it should not be minimized. It is part of really the whole triangle of, I have a problem, I can use these ideas, so I have ideas to solve this problem. But if you don't have a sort of good implementation and something, the aspect of bridging both the sort of innovation and the sort of the impact, you're not gonna get there and you're not gonna make an impact.
I really like the aspect and the emphasis that both of you place on practicality and continuity. I don't think innovation, to both your points, is not a one and done process. It's about learning and using those learnings to actually inform and to grow. So thank you for your great definitions. So my second question for you both, imagine that you've done quite a lot in your career. Obviously, that's how you end up with a very important role at the NIH. But what would you consider to be your most significant professional or scientific accomplishment prior to your current role at NCATS? And we're going to start with you, Sam.
Sure. And so this is before my time at NCATS.
Yeah.
So I would say, back on the, my background is really automation engineering. I'm a biomedical engineer, but automation and industrial engineering. And so I would say what I've been proud of over time has just been able to make things operational. And this is from consistent, one of my very first jobs was working in fiber optics, because when I, came out of school in the mid-90s, fiber optics was booming. And I worked for a company and as a contractor, and we were doing essentially stress testing of fiber optic systems, and so it's been satisfying over the years to, we have Netflix and so forth today because we solved the bandwidth problem 20 plus years ago. But that was, a research project that became, how do we implement it, but then how do we do that test engineering to make sure that those racks essentially are completely functional. And then I did a lot of work also with defense, in some cases really focused on data acquisition systems and data collection systems. Immediately prior to joining NCATS back in 2005, I was working as a contractor at Northrop Grumman, and we were working on electronic warfare. And so those projects and those systems are essentially used, in F-18s now. And so it was also, how do we implement and operationalize that these units that are going to these aircraft are actually, still flying and flying for a prolonged period of time. So I'd say my overall achievements I'm proud of are developing or taking hand in being part of teams that develop systems and platforms that have a long-lasting operational impact. And I think we've carried that over. My heart has always been in science, so when the opportunity came to join NCATS back in 2005, it's been great to apply that to actually making things, run smoothly here, which I think is really one of the most key aspects of my job.
I think your focus and your passion for operationalizing is so important. And I like that engineers like yourself, you have this vision, this unique ability to bring ideas to life, to make these ideas come to life, and to most importantly, think about making things work that will have an impact on people's lives all over the world. So that's a very important thing. And thank you for sharing that. And now switching to you, Mark, what would you consider to be your most significant?
Yeah, so my professional career, it's been really developing biological assays for drug discovery and drug development. So I've worked a lot with people like Sam here at NCATS, but also before at NCATS, I was at a pharmaceutical company. And what was amazing is really bringing both the technology and the biology together, and making the process very efficient in terms of being able to screen millions of compounds, millions of wells, and really making the process very efficient and very cheap, right? And I think one of the things coming up to NCATS was meeting people like Sam and chemists and other groups, and really having these amazing capabilities and automation capabilities, and really thinking about how can we use them to get medicines faster to the patients, right? So one of the platforms that I'm actually very proud, and it wasn't really a new idea, but one of the things that I think highlights NCATS and the work at NCATS is that it brings all these people with different expertise and works together. And what we did was to create a platform, a screening platform for combination therapies. And, there's a lot of therapies for cancer, for infection diseases, there are actually not one drug, but multiple drugs, right? And normally those have been developed hypothesis-based, sort of trial and error. So the goal, again, was what is the problem, right? Well, the problem was, can we develop these combination therapies faster and more efficiently? So with Sam and other people at ANCADS, we put together, with the infrastructure that we had, and some inventions in terms of coding and software and instrumentation, a platform that I believe it's almost unique in the world, in the sense that allow us to screen a lot of pairwise combination therapies. And actually, it's been a very successful platform in that it has been able to inform clinical trials for several cancers. So it is a platform I'm very proud of, not only developing, not only making an impact, but really leveraging people like Sam, the engineers in the lab, the chemists at ANCADS, and informatics to really come up with a platform that is really making a huge impact in finding new therapies.
I really like what you've said so far. I think that the idea of innovation is not one that you can really do on your own. It takes a team, and it takes people that have different types of skill set: chemists, biologists, engineers, physicists. Like, think about it. And that's really how we can get to operationalize great ideas. So thank you for that. And I'm going to switch it up a little bit and start with you this time around, Mark. What inspired your interest in 3D bioprinting?
We started working bioprinting not because of the scope being regenerative medicine. Really, what we were interested is in developing assays that would be more predictive of drug responses in clinic. So as you know, like the process of drug development, it's very, in a way, it's very inefficient, right? When 10%, on average, 10% of the drugs that go into clinical trials make it into approved drugs and into patients, right? So going back to the ANCADS mission of really catalyzing the efficiency of the drug development science to try to get the drugs to patients faster. The question is, like, that's the problem, right? How you make the drug discovery process more efficient. And one way to do it, and it's not our idea either, is to, these drugs that went into clinical trials, they all went through and were active in a cellular assay and an animal model, right? And the fact that then they didn't work in humans, it means that these assays are essentially not predictive of what happened in humans, probably because they are too simplistic. So, so the idea is then, well, can you develop assays that are physiologically, what we call physiologically more relevant, assays, cellular assays that look more like cells in a tissue in a human. And that's really how we got into 3D bioprinting. We got into 3D bioprinting through the development of what people call 3D cellular models, because it uses cells that are in contact with each other. It uses different cell types, which are really what's found in tissues. The hope is that these assays will be more predictive of the drug responses in clinic. And really, as you probably know, there's different type of cellular models, right? You probably heard about spheroids, you probably heard about organoids, you probably heard about tissue on a chip, which is another program that ANCADS have really supported a lot. Our approach was to use biofabrication techniques, and bioprinting is really a biofabrication technique, using tissue engineering techniques to make not big tissues that will be used for regenerative, but actually make a small tissues that you can put in a multi-well plate so you can use for drug screening. And that's really the goal of what we're trying to do. And biofabrication and bioprinting being one of the techniques used in biofabrication allows you to really control very well what cellular complexity you want in your tissue, what spatial and morphology you want in your tissue. And because you do it in a multi-well plate format, you can use the automation capabilities developed by Sam and others in the group to really use the same sort of assay technologies that you use in 2D, apply them to 3D, which is not trivial, but that's the goal, right? You're gonna have these small tissues in your wells, and then you're gonna like test compounds to see if you can correct disease phenotypes in these small tissues.
That is incredibly exciting. I started out my career as a scientist at Pfizer, moved on to UConn, worked at Yale, and I used to imagine the day where we would have a more efficient way of running assays. So I don't have to keep repeating myself with those 96 well assays or even the HTS screening. It was just never seemed to be as efficient as it should be. So I love the idea.
One of our goals is to do 3D cellular models in a 96 well plate. For a 3D screener, 96 well plate is like, what are you doing? That's all new. But for us, it's new.
Oh, that's good to know.
Our argument is that we will get better information, and it's not so much about throughput and numbers. It's really about the information that you get from these assays.
Well, that's very exciting. I think that how you go from large scale to smaller scale, and there was a point you made around customization. I think that that's really the power of what you're doing and the wave of the future. I'm curious to see, for you, Sam, what inspired your interest in 3D bioprinting?
Yeah, very similar to Mark, and I think, he's touching a lot of the points, but especially it may be lower wells, but the complexity is just through the roof. And I think that was part of part of what drew me to it, because we had done some experiments, like Mark mentioned, there's different types, and I'm sure you're familiar with a lot of folks doing screens with spheroids. And so we had done some spheroid screening and even some comparison of saying, okay, the same cell line as a 2D plane versus a 3D spheroid, and then can we see a difference in the compound, dose response? And the answer was yes, but we couldn't quite quantify, is it better than the other? Is one better than the other? But it was kind of fascinating to me just seeing the results that just, again, the same cell lines just prepped a different way, and your drug response is different based upon a 2D model versus 3D. And so the complexity factor was a big draw, but also, again, we had really shown this operational efficiency that we could screen these day in, day out. But then, are we having things hopefully at some point reaching the state of an IND? And the answer was, and look, let's be real, these are tough problems. This is, a tough field that we're all in, is the likelihood. And I think that's one thing we try to prepare all the biologists for. You have to be resilient, because the likelihood is you're going to fail. That's not because you're not you're a bad researcher. It's just the nature of the problems that we're tackling. So part of it was a challenge aspect, but then part of it too was just, if we want to introduce in this 3D space, it was kind of interesting at NCATS, is originally this came to my group because it was automation, because they felt, well, it's a 3D printer. And I was kind of fascinated by this. I said, look, I'm interested, but this is not, the 3D printer is one of the easiest aspects of this entire problem, right? This is one of the most complex systems biology, material science problem I've ever encountered in my life. And we were able, thankfully, to recruit and rope Mark into working on this, who had a true passion for developing, Mark said, is these complex assay systems. And so part of the draw was, one, we were doing something, we're doing it well, and we weren't really seeing better results. And that spurred this need for innovation. But then it was also, and back to the point that way NCATS works in teams, how do we recruit the right team of the right people to really get this to a point that we could implement this properly? And like Mark said, it is kind of interesting where we're kind of going back to, we were proud of 1536 screening, but now when we get that first, and we've had several 96 but again, tissue constructs, just the complexity to produce those, and then the complexity to measure those is just through the roof. And I think that's also the challenge aspect of it, is what's been really exciting about this project. There's multiple challenges across the way, and I think those are the fun problems to work on, because it's not easy at any step.
Yeah, but I think the challenge is being able to find opportunities and challenges is really what innovation is all about. And it shows sort of the strength of your team at the NCATS, that you approach these problems in a multifactorial and multidisciplinary manner. So that's very exciting. Now, I'm curious, obviously COVID-19 has impacted pretty much every industry. It's impacted us personally, psychosocially, financially, emotionally, etc. How has COVID-19 impacted your role, Sam, as the Chief Information Officer and Director of the Automation and Compound Management of NCATS? And do you have any involvement in the National COVID Cohort Collaboration, N3C?
Oh, yeah. Well, so a couple things I would say. It's great, a little bit off of tissue printing. So one, I felt my job as a CIO is to make sure everyone can keep on working remotely to the best of their ability, right? You know, and I think, you know, at NCATS, we have an amazing support team when it comes to making sure people have all the equipment, you know, the basics, laptop, connectivity, Zoom, you know, every tool imaginable. We have, you know, OneDrive, we have Box, we have Teams, we have G Suite. So part of it was just making sure that the workforce, if anything, complaint we get is that we have too many tools, to be honest, but I'd rather be in that position that we have too many tools.
Yeah.
And so in our role, it's impacted that. For N3C, what I would say there is, you know, my role, we actually started, the reason we were able to respond and put N3C up so quickly, and you know, just at this point, we're over 2 million plus patients in, 300,000 COVID positive, 2 billion rows of data. Last week, we actually had our 600th researcher, you know, essentially sign up for this platform. And so, you know, my role of the CIO is we map this out years in advance. And this gets back to something that I think Mark and I have talked about is how do we develop these platforms that you can operationalize and then pivot the need to. And I'll let Mark talk about what they did on the tissue printing side and all the COVID work they're doing, because, and this also ties to something Mark mentioned about the combination screening platform is, you know, Mark mentioned we've built this capabilities in-house, but what else is beautiful is we can actually send these plates with compounds to collaborators around the country. So we haven't just operationalized it for ourselves. We've operationalized it for our contributors and our collaborators. But on the N3C side, so my involvement, I would say, is really behind the scenes, and I view our group as a foundational piece, is we need to build a secure data enclave that's going to take these electronic health records from data contributing sites and provide an analytical environment where folks can start deriving scientific value. And also to date, we have, if you include pre-publications, we have 5 pre-publications in the works. And given that all of this was stood up essentially in early May, to me is just an incredible achievement. But again, we were able to move so quickly because we did the work before to make a platform that we could operationalize quickly. And that's a key point, you know, and so I'm glad you mentioned that, and it's very much in line with the work Mark and his group have done on the COVID side, which is, again, phenomenal, given this all took, you know, like you said, it changed our whole life, you know, going essentially since late February, March, but the amount of work that they have going on there in a relatively quick period of time is incredible.
I think the proactivity and efficiency is remarkable. And Mark, I'll be curious to, you know, learn more about what you all are doing as well as a result of COVID and any involvement, of course, that you have with the N3C.
Yeah, no, I want to follow up on what Sam is saying on the fact that we're very much into the development of scientific and technical platforms. And the beauty of this platform is that you can pivot, like Sam said, to different needs, right? So for the 3D tissue bioprinting, what we're doing is create a portfolio of different, let's call it normal tissues, whether it's skin, whether it's retina, whether it's brain, and whether it's lung. And first what we do is we develop these tissue models. We characterize them, morphology, cellular content, function. And then what we do is introduce something to generate a disease phenotype. So one thing could be cancer cells. So you create a cancer model, but the other thing could be a virus, right? So before COVID-19, we were developing lung models, and we were interested at that time, we were interested in cancer models. So we were very interested in developing lung cancer models. And then COVID-19 hit and said, well, wait a second. We have these lung models that we have developed for cancer, but nothing stops us from taking these lung models and getting viruses and infecting them and using these lung models to investigate SARS-2 infection, or for that matter, any respiratory virus, right? So if you think about it, it's actually a very efficient way to really, from a basic normal tissue that is well characterized, well validated, and we have assays for, and really expand it to any disease that is relevant to the lung with the proper cells and the proper sort of reagents. In this case, we were able to get a hold of some viruses. We weren't able to get the SARS-2 because we don't have the VSL3 facilities, but we were able to establish collaborations with labs that had a VSL3 facility. And like Sam said, they can chip compound plates. So we were able to chip tissues, plates with tissues, and they would do the infection and then fix and with the proper safety measures, send them back and then like do the assays here at NCATS, because one of the other niches at NCATS is that we have the technologies, the assay technologies to do a lot of these assays in-house in a very efficient way. So sometimes it's actually better to get the plates back and do the assays over here. The fact of the matter is, like, we were very efficiently able to pivot a lung model that was for lung, or a lung tissue that was for lung cancer, into a lung tissue that we could investigate respiratory viruses. And one more point is that we're talking about SARS-2, but I'm sure it's not going to be the last respiratory virus that we're going to have around, since they seem to come back in a sort of periodic way. So the idea is that we will validate this platform.
Okay.
And the hope is that we'll be ready for the next one. So when the next virus comes, we'll be able to then take the virus, investigate it in these platforms, and actually use these 3D cellular assays to find compounds that hopefully will be more efficient.
Yeah, I mean, your responses, I don't know if both of you talk the same way all the time, but I feel like I'm talking to two mirror images. It's like there's such cohesion in what you say. And I think that that's a testament to the organization that you're both a part of. I think the mission, the objectives are very clear. The approach is to be as forward thinking and proactive as possible. If you can anticipate issues before they happen, then you can blunt some of the challenges that you would have with that. So very well said, both of you. I'm very impressed with the work that is being done so far.
Thank you.
Yeah, my pleasure. So my next question is for you, Sam. 3D bioprinting has been around for some time, but I think we've started to hear more and more about it in the past decade or so. Do you think we're ever going to get to a point where it's sort of a mainstream technology that you'll find in your, average academic lab or anywhere that you can imagine that research is being done? And what are some challenges as we think about sort of a widespread mainstream application of 3D bioprinting?
It's a great, one, I would say, I think it's already approaching the academic space. And part of it just comes, from that investment, in technologies. And I think that's what, the past several years in tissue printing, what it's taught me is that, we mentioned high throughput screening. And if you look at high throughput screening over multiple decades now, really, you can say going back to the early 2000s, late 90s. So it's, we're close to 20 years. There have been billions of dollars of money invested in all these sort of support and foundational technologies to fill in the gaps, to put us in a position that we can screen, a million plates, like I mentioned. And I think what we've been noticing on the tissue printing side is we're just now starting to realize what some of those pain points are. And there's also investment in terms of going into those foundational technologies to make all of it work. And I think when you start investing more in that space, the cost is going down. And I'll give you an example of this even on the screening side. If you had said 10 years ago, you need a high content imager, you only have 2 or 3 choices. They're sort of niched instruments, and they have their strengths and weaknesses, and they're all very expensive. And I think what's phenomenal over time with investment is the cost of some of this instrumentation and some of the equipment is going further and further down. And so you see some of this now in the tissue printing side from a commercialization perspective, as there's companies out there trying to make instruments, they're trying to make bioinks, they're trying to make reagents. And I think, in all of these, it's really like, I view science moves almost, that's why it's funny also you mentioned, innovation, back to the very first point. Innovation to me happens almost a micron at a time, right? And it's almost like you have parallel measurements of these different microns at a time, making these small incremental steps until they're measurable. And so I think, and even one basic example is there's plenty of academic labs now doing awesome assays, and their first foray into this space might even just be with spheroids, or layered systems, right? You don't even necessarily need a tissue printer. And so I do think that we're going to reach a point that's becoming more and more prevalent, but it just takes time. But I do think also, a point Mark made going back to leveraging off of the existing infrastructure where folks have already invested, right? That's why, again, you mentioned a 96 well plate, and you'll probably never escape a 96 well plate, but billions of dollars have been invested to make sure that there's a lot of different things you can do with the 96 well plate at this point. And I think as there's more investment along this area, I do think we'll reach a point that'll be much more prevalent in the academic side, because let's be real, for a lot of the labs, we're fortunate at NCATS, we have resources that many labs could never dream of. And that's why we try to work with those collaborators to give them access to resources that they don't. But I do think it's going to be more prevalent and cost effective over time.
And what's a possible timeline in your opinion? 5 years, 10 years, 15 years, 20 years?
I feel, again, depending on the metric of success, I would say 5 years is reasonable because, again, we already see movement in that direction. You know, I think we have a lot of academic groups now that have reached out, you know, to work with Mark and some of the collaborations that he has. I don't think it's too far off, to be honest. And again, there's players getting into this space now. You know, a lot of folks are really pushing, you know, to bring the cost of some of these foundational pieces down, you know, such that labs can afford to do it. Because that's a big part. And I think that's something, again, at NCATS, we try to spur any way we can is ultimately have the best ideas, but if people can't afford the means of implementation, it won't be adopted, you know? So I think we have to figure out as a community how we make a lot of this technology affordable, and the affordability will directly relate to the adoption.
Great.
I feel
and
5
Mark,
years.
5 years? Okay, great. Now I'm curious to know what Mark thinks about both questions.
I think one of the biggest challenges is going to be managing the hype. And because it is a very appealing technology, right? I mean, it started many years ago with regenerative medicine, making tissues for really transplantation. And what we're saying now is like, wait, you can use these technologies also for making tissues in a multi-well and using them for screening. And you can use them also for investigating biology and what we call disease modeling, right? And I think one of the biggest challenges is in each one of these areas, there's both a strategic challenge and a technical challenge, right? I think this the strategic challenge that we have for us in drug screening is what a lot of people are asking: show us data that indeed these 3D models are more predictive of clinical drug responses than 2D models, right? So people are asking for the data, which is, look, it makes sense, right? So designing experiments to prove that really when you test a compound in a 3D model, the data that you get is actually better in any way than 2D models, whether because in a 3D model, you can recreate a clinical biomarker that you cannot recreate in a 2D model, and that you can come in with a drug and modulate this disease biomarker, which is really what you want to do. That's one of the challenges for us, right? And then when it comes to technical challenge, Sam is totally right. These bioprinters, some of them are getting very affordable, to the point that an academic lab, whether it's individual lab or core facility, can have these bioprinters. The aspect of really learning when to use a bioprinter, when using a bioprinter makes sense or not because you have a hammer, everything is a nail, right? So not because you have a bioprinter, you want to bioprint everything, every tissue, every 3D model. I mean, sometimes it makes sense, sometimes it doesn't make sense. And sometimes you use a bioprinter because you want to create a pattern that helps you develop an assay and quantitate some biology better. So it's not necessarily obvious, but it's actually an application that we've used a lot. And it really has helped us a lot, not making the tissues, but being able to quantitate biology on the tissues. And that's an aspect that it's very often forgotten. It's not enough making a beautiful tissue and doing the histology to validate it and showing that you have to on the right cells and the right function. But now what, right? I want to develop an assay that I can use to screen compounds and demonstrate that a compound can correct a disease phenotype. So developing assays in 3D cellular models is quite challenging. Yeah. And that's the only reason that Sam is also involved, and because we generate a lot of data. We use fluorescent imaging a lot. We use confocal imaging. We generate tons and tons and tons of data. So you need the infrastructure to be able to keep up with this data. And that's not trivial. And one of the biggest issues that we have is that we generate so much data that it essentially, that the whole transfer of the data, storage of the data becomes a bottleneck.
Even with a 24-well plate, but as one example of that, we literally just ran into this last week, and this is also, Mark has an amazing team, but they have a 24-well tissue, but then they're doing an 11 by 11, essentially number of fields. And so 121 fields of 60 Z stacks and then 4 channels, right? So you do the math, right? So 24 times, 121 times 60 times 4, and that's one plate. And then, to Mark's point about designing an assay, ultimately you want something measurable, right? And I think that's the key point about all this. And so, yeah, the pretty pictures are one thing, but you want to make a measurement to determine what you're doing scientifically next. And so, these problems are something that's, part of the reason as well. My title now is CIO, but really it was a preemptive strike because science is becoming an IT problem. And I thought, we need to be in a position that science will tell IT what it needs to do. When we do our best, but again, Mark makes our job difficult because I think at this point I've lost track of how many high content readers and microscopes we have, and then Mark has a small army of incredible researchers just churning out experiments. We're doing our best to handle how that scales because there's some real challenges with it just by the sheer volume of data we're talking about, right? We are, in the terabytes and soon to be petabytes and growing, right? Which is exciting. Again, this gets back to exciting problems that we like solving those, but again, for a scientific output and a scientific need.
Yeah, I mean, I think it's very well said. I personally believe that a data-enabled patient is probably going to be the main focus for the next few years because to your points, both of you, we're generating so much data. How do we make sense of that? And in real time too, so that we can inform decision-making. Mark, I just want to do a quick follow-up with you. What's the timeline that you think before we can have sort of prevalent use of 3D bioprinting in most academic labs?
Oh, no, I think that's a reality. I mean, 5 years, I think now these printers are very affordable. They have a lot of capabilities. I think, these companies are growing. So it is a reality now that academic labs are purchasing this technology. And then what's going to be interesting is how they use them. My hope is that there won't be a wave of excitement and then kind of like learning how to use it or not. And then the bioprinter will be growing dust somewhere in the lab because you need so many cells, you need the technologies, you need so much that whether it makes sense or not for whatever application the lab was planning to use it, that's going to be the big challenge.
No, go ahead, Sam.
No, I was going to say it's interesting. That's one thing that Mark mentioned, and I'm glad he touched on that point. We were able to pivot to 3D printing because when we're running 1536 well plates, the sheer volume of cells required wasn't daunting to us. If we're doing a 1536 well plate and every well has to have 500 to 1,000 cells, we're used to that large capacity tissue culture. There's not many places that can generate that many cells. That in itself becomes a real daunting. There's some pretty basic, challenges to all of this. Is one, can you grow enough cells, and can you make sure that the tissues you print can actually stay, contaminant free for months on end potentially? Not trivial. There's a lot of good labs out there, but again, there's some real logistical problems. I just want to touch on that one point. Is the cell production is a real problem because cell production still, as much automation as we bring, comes down to biologists coming to the labs at all hours of the night. So I'm truly amazed by their dedication because in many cases, if you're handling multiple cultures for large volumes of cells, you're on the cell's clock, so it's really a seven day a week operation here at NCATS just for the cell production itself is that's also a key factor of what's going to limit potential adoption.
And for us, because we want to make the tissues more physiologically relevant, we strategically decide that the tissues that we're making will be made with primary cells, human primary cells or human iPS-derived cells. So obtaining these cells, growing these cells in itself it's a logistic endeavor, and it's not easy and not trivial. I mean, it's a little like sort of not much of making sense if you're going to make these complex tissues and you're going to use cell lines, right, that have been passaged for years and years. To us, it doesn't make sense make these 3D complex tissues with HeLa cells or with educate cells. No, we want to make them with cells that are relevant, whether it's primary or iPS-derived cells.
Well, I think that that is great. The points that you both have mentioned, I actually like that you highlighted the challenges as well, because wherever there are opportunities, as we've talked about, there are challenges as well. So just thinking through it all, what are some key consideration factors that you think will be important for sustaining innovations like 3D bioprinting in the life science industry?
Well, that's one of the reasons that I came in NCATS,
Yeah.
right? The emphasis on both innovation and giving, and that means giving resources, right?
Mm-hmm.
Giving resources and sort of funds and people to do sort of the novel, the aspect that you don't know what's going to happen, right? But if it works, you think that it's going to make an impact. And then at the same time, having at NCATS under the same roof, a whole platform of screening, of probe development, of drug developments, and being able to sort of leverage both. So, so really, we don't want to work as a 3D group, bioprinting group. We don't want to work in isolation, but we want to be given the time and the resources to make these models, validate these models, do proof of concept, and then going to our sort of colleagues that are developing probes or developing drugs and say, look, we have these models, these 3D models, and we think that it's going to help you develop better drugs and pick better compounds for medicinal chemistry. Let's give it a try, right? So we are very mindful of both the aspect of innovation with your own timelines, but with an eye on the other aspect of what ANCAT is doing, which is probe development, drug development. So because we need to integrate the both, but at the same time, it's the milestones are different. The metrics for success might be different, but we need to be able to integrate both and have both going and funded and resourced.
I couldn't agree with you more. I think it's challenging to think about how you can come up with this sort of outside the box ideas, but still make it practical and feasible and fit within a time frame, all that stuff. So I completely understand that. And Sam, the same question for you. What are your thoughts?
Yeah, no, I think and Mark's highlighting a lot of this about, the key aspect of what we do at NCATS also is dissemination. And I think, for this to last and be sustainable, I do think we're going to need more collaboration, because a lot of this is, I would still view, somewhat pre-competitive. Like, the people want to publish on their different printing techniques, but I think one thing we've seen over time, which to me has been fascinating, is when we first got started, we had two primary projects. We had skin, and then we had a retinal tissue, and we weren't sure if the techniques used there would be transferable to other tissue models, and then sometimes they are, and sometimes they're not. And I think a key aspect we've learned over time is, it'd be great is, how do we share information, both what with worked, but also with what didn't work. And the reason that that's an incredibly important one on this is the time it takes to grow that number of cells, the time it takes to grow your tissues is the iteration, per experiment is in some cases months. And if the community can share with each other of what works and what doesn't work, I think everyone will advance together. And again, we're already starting to see that, with different labs and, sort of the exchanging of protocols. So I think long-term, for it to succeed, and long-term for this to really get adoption, is just that continued, the labs getting funded, labs trying this out, but then also labs communicating and collaborating. Because I think it's going to be a huge, factor for this to really gain adoption, and ultimately to prove it, the jury's still going to be out, right? It's ultimately going to say, that's our premise, is that we think we're going to have, more physiologically relevant models. Can we prove that, to show that the value is in adopting these platforms, because there's quite a bit of work involved to get it set up. But I think that long-term community sustainability is going to be important. And it's been great to see, because, we mentioned some of the manufacturers, some of the vendors, from equipment into these tissues. There's a lot of user groups being set up, which I think are great, so people can sort of share the best practices. And I think for the sustainability, that's going to be a key to this, because it is a lot of work. And I do think this is where sharing of information could really benefit the entire community. Because the iteration per experiment, again, that's why I have a lot of respect for the work the biologists do. They might have to wait between cells in their tissue 3 months per experiment to find out something that didn't work,
Right.
which is far different than if you're running some of the 2D experiments, right? You have multiple cultures running up, and you can run it in the 2D plate and quickly iterate. You don't have as many iterations. So the more we can share information, the better.
Great. And I think to the point that both of you made earlier, resilience is going to be key to all of this. If you're not really committed to doing the work, we're not going to see actual results. And we have to have that continuity in the thought process. So in closing, I would like to give both of you the floor to share any final commentary or thoughts that you might have. And maybe I'll start with you, Mark.
I had a postdoc in the lab, and she was working on skin models, and she was doing some experiments, and she was comparing actually 2D cultures of the cells versus a 3D skin tissue. And the data that she was getting was just amazing, right? Day and night, right? That she was able to recapitulate clinical biomarkers in the 3D model, and she was able to see the effect of compounds. And she told me, it's like, I just, after seeing these data, I don't know why anybody would investigate biology in 2D with cell lines. It's just so different when you have the relevant cells in a tissue, the relevant physiological complexity. And to me, that really stuck, because we already having evidence that when you start doing 3D biology in 3D, it's different from 2D. And it's closer to clinical, which is exactly what we want to see. And like Sam said, that was the hypothesis, if you wish, right? So we're starting to have quite a bit of evidence that when you do biology in 3D, is more clinically relevant. The effect of the compounds really matches what you see in clinic, and we need more of that. But I think little by little, sort of through development, through integration in drug development pipelines, through collaborations, like Sam said, academic, industry, even regulatory agencies, I think we're all very interested in sort of being able to demonstrate that these tissues will be really more predictive.
Well, thank you for that. I like the focus on improved efficiencies. And I think through data sharing and perhaps educating the public through forums like this, we can continue to increase knowledge and awareness of 3D bioprinting. Now is your turn, Sam.
Yeah, no, everything Mark said, but then also just, again, the focus on the platforms. And, it's great that you mentioned N3C, because that right now is a clinical informatics platform that has EHR data on patients. And what else we're doing now actually is we're actually ingesting, based upon the Cancer Imaging Archive, has publicly available data of radiology information from COVID patients. And so now, Mark, we were mentioning all the images that Mark is producing for tissue printing. Is now we're focused on getting also imaging data in for COVID patients and actually having that as part of the overall enclave to match with the electronic health record. And the reason I bring this up again is all these platforms to me are very related, right? So the platform that we're creating for Mark to store images and also to essentially, be able to make measurements, we're going to use that same platform, those same techniques to actually get images now for, COVID patients, both radiology, but also pathology. So it's interesting how there's an overlap I see, because, a standard that Mark is still using on the tissue side is still histology, right? The tried and true H&E stain is exactly what they're using for COVID patient histology as well. And then how do we build these foundational pieces at NCATS that we can actually bring this data into collaborative platforms and then give access to it for research, hopefully to drive discovery. And so I think, the focus I'd like, to end with is just, I view what we do at NCATS as develop these platforms. It's really been an honor and a privilege to work with folks like Mark to drive these platforms forward. And I just view our job is to make these platforms available, not just to NCATS, but the scientific community as a whole, so we really believe that, again, we're the government, we're taxpayer funded, and we just want to make sure that, the tools we're developing aren't just for us, it's for the scientific community at large. But it has been really interesting seeing that these platforms in some ways are kind of merging in certain areas, as now we're start talking about all the challenges of acquiring this imaging data. And it's been also the scientific response has been incredible about people supplying images. There's any silver line to COVID, it has been the scientific community rallying and actually finally willing to share, right? I mean, so it's so what I would just say again, the highlight I would just end on this is just we build platforms at NCATS, and we're really excited to build more, and we want to make sure we build platforms with the scientific community as a whole, as really our customer, and to make sure we're serving that customer well.
Well, thank you both. I know anyone that watches this video podcast or listens to it will be blown away by the wealth of information that you've both displayed. And it's made me personally very optimistic about a technology that I'm already optimistic about, which is 3D bioprinting. I always sort of put it in the 5-year, 15-year timeline, somewhere in the middle. But I think that from what you're both doing, I think the future looks great, and thank you for your commitment to transparency and sharing knowledge as well. I think you've demonstrated that by being a part of the Amplifying Scientific Innovation platform. So I appreciate both of you, and I look forward to staying in touch.
Thank you so much, Sophia. Thank you for having me.
Thank you, and have a good day now. Okay.
You too. Bye-bye.